Sharks have hunted the oceans for over 400 million years, longer than trees have existed on land. In that time, they've developed a sensory toolkit that puts almost every other predator on Earth to shame. The ability to detect a single molecule of scent diluted across a swimming pool, to feel a heartbeat through open water, and to sense electric fields too faint for any instrument humans carried until the 20th century. So what actually draws a shark toward a source of food from hundreds of meters away? Is it blood, movement, sound, something else entirely? Today, we're running a structured comparison of 6 different signals side by side to find out which ones sharks actually respond to first, and busting a few myths on the way. This is the shark bait experiment. Understanding what draws sharks towards a stimulus isn't just curiosity. It has real consequences. It shapes how beach safety systems are designed, how commercial fisheries reduce accidental shark bycatch, and how researchers safely attract sharks for tagging and population studies without disturbing their natural behavior. For decades, most of what the public knew about shark attraction came from a mix of half-remembered biology class facts and dramatic retellings. The idea that a single drop of blood could be detected from miles away being the most famous example. Some of that turns out to be true. A lot of it doesn't hold up once you actually test it side by side in controlled conditions against a shark's real sensory range. A shark's sense of smell begins in a pair of nostril-like openings called nares, located on the underside of the snout. Unlike human nostrils, these have nothing to do with breathing. They exist purely to sample water flowing past the shark as it swims, funneling it across folded tissue called olfactory lamellae. Roughly 2/3 of a shark's brain by volume is dedicated to processing smell, and their sensitivity to certain compounds, particularly amino acids released from injured fish tissue, is extraordinarily high. But extraordinarily high is not the same as the popular claim of detecting 1 drop of blood from a mile away. In reality, detection depends heavily on concentration, water current, and the specific compound involved, a distinction our experiment is built to test directly. Beneath the skin around a shark's snout sits a network of small jelly-filled pores called the ampullae of Lorenzini, one of the most sensitive electrical detection systems found anywhere in nature. These organs can detect electric fields as faint as a few billionths of a volt per centimeter, sensitive enough to pick up the tiny electrical signal generated by another animal's muscle contractions, including a heartbeat, through open water and even through sand. This sense becomes most important at extremely close range, the final few centimeters of an attack, once smell and vision have already done the work of bringing the shark close. It's also why sharks occasionally investigate objects that give off a stray electrical signal, like a boat's damaged wiring or a research instrument, even when there's no scent or movement involved at all. Contrary to older assumptions, most shark species have quite capable vision, adapted for low light, with many able to see in color, and some possessing a reflective layer behind the retina, a tapetum lucidum that dramatically improves night vision, the same feature that makes a cat's eyes glow in headlights. Running the length of a shark's body is the lateral line, a row of fluid-filled canals just beneath the skin, lined with tiny hair-like sensors that detect changes in water pressure and low-frequency vibration. This system can pick up the irregular, struggling movement of an injured fish from a considerable distance, often before smell or sight ever come into play, making erratic vibration one of the very first signals a shark actually registers. To compare these signals directly, our team set up 6 identical release points, spaced evenly along a controlled offshore test line, each rigged to release exactly 1 signal type at a measured, consistent concentration: whole blood, a concentrated amino acid solution, fish oil, a low-frequency vibration source, a moving decoy with no scent at all, and a weak electric field generator with no scent or vibration. Each release point was monitored by remote camera, with response time logged from the moment a signal was released to the moment the first shark oriented toward it. No bait was ever attached to a hook, and no shark was fed, chummed at close range, or put in contact with a person at any point in the test. The whole blood release drew the fastest average response of any single compound signal, with sharks reliably orienting toward the plume within a few minutes at close range. But the response dropped off sharply with distance and turbulence speed, directly contradicting the "one drop from a mile away" claim. In real ocean conditions, turbulence and dilution break a scent plume apart far faster than the myth suggests. The concentrated amino acid solution, designed to mimic compounds released from an injured fish's tissue without any blood at all, performed nearly as well as whole blood, and in low current conditions actually outperformed it, supporting the idea that it's specific chemical compounds, not blood itself, doing most of the work. The low-frequency vibration source, mimicking the erratic movement of a struggling fish, drew the single fastest initial response of the entire test, with sharks investigating within seconds, well before any scent plume could have physically reached them. This matched the lateral line prediction closely. Vibration is often the very first cue, not smell. Fish oil performed the weakest of the scent-based signals, breaking down and dispersing faster than the amino acid solution. The scentless moving decoy drew occasional investigation, but far less consistently. The electric field alone drew almost no interest from a distance, but nearby sharks that approached for other reasons were repeatedly observed making a final close-range correction toward it in the last few centimeters, exactly matching what the ampullae of Lorenzini are known to do. Ranked by average response time across every trial, vibration first, amino acid concentrate and whole blood close behind and roughly tied, fish oil next, the scentless decoy after that, and the electric field alone drawing almost no response until a shark was already within very close range. The biggest myth busted, it isn't smell alone, and it certainly isn't a single drop of blood from a mile away. Real shark attraction is layered. Vibration and low-frequency sound do the early long-distance work. Scent narrows the search and holds a shark's interest as it closes in. Vision takes over as it approaches, and electroreception makes the final precise correction only once the shark is essentially on top of the source. No single sense does the whole job, and no single myth captures how the system actually works. Understanding this layered system has real practical uses. Several modern shark deterrent technologies work by disrupting electroreception at close range rather than trying to mask scent, which, based on what we just saw, makes far more sense than older approaches ever did. Fisheries use the same knowledge to design gear that reduces accidental shark bycatch, and researchers use carefully controlled, low-concentration scent trails, not blood, to safely bring sharks close enough to tagging and population studies without disrupting their natural hunting behavior. Sharks aren't the indiscriminate hunters they're often shown to be. They're running a precise, layered sensory calculation with every approach, built over hundreds of millions of years and refined trial after trial, long before any of ours. Subscribe to Abyss Report, Ocean Science, every week.